Yeah. Do you want to start?
Yeah. So I'll start sharing my screen. Oh, you have to allow me.
Let me give you privileges. You're a co-host now. Do you
All
have
right, great.
access? Great.
Yep. Okay. Can you see the notebook now?
Yep.
Okay, cool. So hello everyone again. So I would like to make this. Wait, I can't see the chat like that. I first have to go back here. So I want to make it as interactive as possible. So whenever you at any point feel like something's unclear or you have questions or whatever, just feel free to interrupt me. I wanted to do like a little mini questionnaire just to know how much of experience you have with the topics being covered. So could you in the chat like type one if you already did any kind of analysis using a TRF or like a TRF model? It doesn't matter if it's in Python or MATLAB. And two, if you never did any TRF analyses yourself. Okay, so kind of mixed here. Most people already have done some kind of TRF analysis. And then again, the same thing, maybe type one if you feel somewhat confident in using Python, and two if you feel like you have very little experience using Python. Okay, seems more twos for that answer, I say. So yeah, just so I know that I don't like spend too much time on extremely basic things. And but again, if feel free to interrupt me at any time. And so I'll get started now. Oh wait, I have to share. All right. So as I already said in the talk, we'll be working with this data set, The Old Man and the Sea. I recently uploaded that to OpenNeuro. So if you've never used OpenNeuro, it's really cool. It's this online platform where people share data sets. All the data sets there are in the BIDS format, which is a standardized format for storing data. And the whole thing interacts with a software that's called DataLad. And who hasn't done it yet, please execute the code here in all those sections up to section number 2, because that will start the downloading of the data, which takes a bit. So DataLad is a really cool piece of software for kind of managing data sets and analyses. I don't want to dive into the details in this talk because it's a very deep piece of software and it takes a while to understand. But I encourage everyone to have a look at it. I think it has made my life as a researcher a lot better. I never now do any research projects where I don't use DataLad. So definitely take a look at their online documentation and see if it is for you. For now, we just needed to download the data from OpenEuro. So here in this first cell, we're installing some software, which is we're installing first DataLad, and then we're using DataLad to install the dataset from OpenEuro. So this is the address of the dataset. And you can see, like, if I execute that section, it runs really quickly. It's only like 3 seconds. And of course, in 3 seconds, you're not downloading a whole data set. But instead, what DataLad does is it loads the whole structure of the data set. So we see here they're like all the folders, like 19 subjects in total, a folder with stimuli and everything. So it downloads and if you look into those folders, you will actually see all the files for all the subjects are there, but those are not the actual files. Those are just symbolic links. And what's cool about this is that it's a way where you can very quickly get the whole data set and just work on parts of it, and then push back your changes to your original repository. And that way you can still like kind of preserve the structure, and you don't have to download the whole data set. This is really useful if you work with stuff from, for example, human connectome project, where of course you can't download the whole data set on your notebook drive.
Oh,
Yeah.
do you mind increasing your font size? We have that
Sure.
comment from the chat.
That better?
Is that better on the chat? Yeah, let's go with, yeah, great.
Great.
Good.
Yeah, I can't see the chat, so if you can just
I'll manage
unmute
that.
and tell me, or you tell me. Yeah, thanks.
Yeah, or unmute yourself.
And so if you want, if you're not familiar with how the BIDS data structure looks like, you can just take a moment to look at the content of the different folders. So you can just use this command ls with some folders. So I've called the folder here OMATs, so for Old Man and the Sea. And you can list out contents by, for example, saying something like OMATs subject. Okay, you're even getting the suggestions, and you can see each subject folder has only a single subfolder called EEG, because BIDS also includes other modalities. It's like a general neuroscience data type. And then if we look in that EEG folder, we can see there's a bunch of files. So this is the brain vision data format, meaning that for each file there's a EEG file, then there's a JSON file, and then these like VHDR and VMRK. So this brain vision data set like splits the data across multiple files, and then for each file there is also a file called channels, which lists the bad channels in that segment. And the data is divided into runs. So each one run here means like one just continuous presentation of data. And then here, which by now hopefully has finished for everyone, we're actually loading the data. So this is with DataLed. You can say there's a command called get, and you can call this get command on any folder, and then DataLed will check if the contents in that folder are present on your machine, and if not, download them. So here I selected the subject 003. You can use a different subject, so you can just change this number and you will be able to do all the analyses with the data of a different subject if you think that might be more interesting, if you're feeling a little adventurous. Of course, then it might be that you pick one of the subjects which have really noisy responses, and you won't be able to see very nice results. So it's a bit of a dangerous choice if you just select at random. But feel free to do that if you want. And then finally, we have to install some Python dependencies. We'll, of course, use mtrf. That's the package we're interested in. I also download MNE, which we have to use for loading and pre-processing the EG data. And then another small package called slab, which is another project I'm involved with, and which is a toolbox for acoustics and sound processing, which here I'm just using to load and pre-process audio. But if you do psychoacoustics, you might want to check that out. I put a link in here. Okay, so did that work for everyone? Anyone who has had troubles downloading the data? No, that's great. So we'll start with a simple univariate model of the acoustic envelope. So probably if you're all familiar with TRF analyses, you know that the envelope is probably the most widely used or most studied feature when it comes to speech processing. There's also a bunch of reviews on envelope tracking studies. I've linked one here, for example. And so first we have to compute the envelope. So here, I think I didn't mention that up here with the get command, we also downloaded the content of the stimuli folder, which contains the WAV files of the presented audio, and also word and phoneme annotations, which we'll not use here, but if you're interested in doing something with that, they're also in there. And so first we have to compute the envelope, and the envelope is obtained by let's maybe first start the code, because it takes quite a while to run. This is, I think, the bit of the code that takes the longest to run of all the stuff in here. The envelope is obtained by computing the Hilbert transform of the data, and then which gives you the analytic signal, and then you downsample that to 64 hertz, because usually we're interested in the low frequent part of the EEG response, and so there's no reason for us to keep all the data to resolve high frequencies. So downsampling everything to 64 hertz will make it a lot quicker to compute. One thing that's really important here is to downsample to 64 hertz after computing the envelope, not before. Because if you downsample to 64 hertz immediately, you'll lose most of the information of the speech signal, because most stuff happens above 64 hertz. And then you'll still get out an envelope that kind of looks like an envelope should look like, but it won't carry any information. And actually, that recently happened to me, where I had some analysis running, and all the predictions were complete garbage, and it took me a whole day to notice that I did the resampling before computing the envelope, and that was actually the issue. So I am downsampling here before doing the envelope, but only to 1470 hertz. So this is and just to like speed up the computation. So this is still enough high of a sampling rate to get most of the interesting stuff in the speech signal. And we can see, okay, it's still running on. So maybe we can already look at the plot. You should be able to see the plot here, right? Or is it just for me? Are you also seeing the plots in your own notebooks?
Yes, I am.
Okay. So here I just pick like a short segment and plot the speech and the envelope, and you can see that the envelope is kind of just similar to like drawing an outline along the speech. And we'll use that for like now the first part of actually for the most part of this tutorial, we'll use the envelope as our predictor. Does it look similar for everyone else? You also have like half of the files processed.
Could you do a little survey with like a 1 if you're processing the stimulus envelope, or a
good idea,
or a 2, or do you want to coordinate that survey? Like, are some people still downloading the EEG? Yeah, that's useful. So is that all envelopes?
Okay, similar to what I'm experiencing. Yeah, maybe we should have also ran that earlier. But I promise the remainder will not take so long. And then after that we have another part that takes a bit. Maybe I'll go over this now. So then after we got the envelope, we'll load the EEG data in section 2.2. This uses M&E Python. Oh, and maybe a 1 if you're familiar with M&E Python, and a 2 if you're not. Okay. Most people haven't used M&E Python. Yeah, so M&E is kind of the most widely used Python toolbox for working with EEG data. It's very nice. I like it a lot. It has great online documentation. So here, what we're doing is we're listing all the files. So I'm not gonna I'm if no one asks about it, I'm not going to explain any like one particular Python command in detail, but rather just kind of explain what they're supposed to achieve. If you have like particular questions, feel free to ask. So we're here creating a electrode montage because our EEG data does not contain information about channel locations, but we'll need that information for the pre-processing. So we just create the standard montage for the 128 channel biosemi cap, which is the one that was used to record the data. And then here we'll just list out all the files and sort them in this line here, and then I'll print them out to see that they're in the right order, because that's sometimes can be tricky, and when you mess up the order of your files, the results can be pretty bad. But here it seems okay, so we have like run 1, run 2. That's why it's important, if you number them, that you don't number them just 1 2, but 01 02, because otherwise the order will be messed up. And then we go through all those recordings and channel files. So the channel files just contain the listings of bad channels. We load the data using these M&E functions. We interpolate the bad channels. Then we filter between 1 and 15 hertz, which is also kind of the standard window people use for envelope tracking. And then we resample the EEG data to 64 hertz, just as we did with the envelope. And then we re-reference the data to the global average. And then finally, we'll get so this is like an instance of the M&E Python raw data class, but we don't need this class. We just want to get the NumPy array inside the class that contains the data. So we use this get data method to just get the NumPy array that's inside this raw object. And then we have to transpose it, meaning we have to flip it around, because M&E has the channels as their first dimension and time points as their second dimension. But most other stuff and the MTRF Pi toolbox uses it the other way around, meaning the first dimension, or in Python it's starts counting at 0. So the 0 dimension should be time, and the dimension 1 should be channels. So we have to transpose it, and then it's just appended to this list. So right here at the top, I'm creating an empty list, and then I'm appending each the raw file, the data matrix of each recording to that list. So let's check. Oh, yeah.
I have a basic question, and then Camila, if you want to unmute yourself after asking the question, you could, or I can ask your question. Just let me know. My basic question is, how difficult is it to take EEG data that you parsed out in MATLAB and saved as a .mat, and then set it up so it can be read, but set it up so it's an M&E data structure format?
That shouldn't be too hard. There is so you can load mat files just in Python. There is the library SciPy has a function that's called load mat. So I think that should be.
Like, is the M&E data structure analogous to a MATLAB structure?
The M&E data, so
like, is it easiest to save a structure as a .mat and then load it in as SciPy? You like
Yeah, I mean, if you're just interested in using mtrf-pi, you don't need to use M&E at all. I mean, M&E with M&E you can use all their like nice pre-processing functions like
mhm.
filtering and so on. But if you don't want to use that, you don't have to use M&E, because in the end, right, we're anyway throwing away the M&E object and just keep the NumPy array.
Okay.
So and I think if you do like load mat, you're also getting out a NumPy array, so you can also um, just use that and then use some other functions for filtering and not use M&E functions. But also in M&E, you can just create a new raw data object from scratch where you yeah, here's like a tutorial on that where it's a bit of a longer process because you have to add like manually add all this meta information like the names of channels and so on that probably are not included in the mat file.
Okay.
um,
Thanks.
so it might be easier to just skip the M&E step.
Camila, would you like to unmute yourself to
Sure.
ask your question?
Yeah, thanks. My question was regarding the reference because I saw you were using the global average, and I was wondering whether for computing TRFs in particular, is there any recommendation regarding which reference to use, or it doesn't really matter?
Yeah, I think the generally in all EEG stuff, the question of reference is like really a tough one, and it's a very deep literature, and I don't know, it has been debated for since EEG is around what's the right reference, and people have different opinions on it. I like to use the average as kind of a default if you don't have a compelling reason not to, because it's robust, meaning it doesn't really like a single bad channel doesn't have a big effect. For example, people often use the linked mastoids, which is fine, but a downside of that is if then one of your mastoid channels is bad, these and has some artifacts in them, that they will project into all the other channels, and this is less of a problem with the average. And then the average is also kind of more fair to like different kinds of signals with different topographies. If you know that you're only looking for auditory stuff, it's fine using a mastoid. But I think the average is like a reasonable default if you don't have any strong opinions about any particular reference.
Thanks.
Okay, so the envelope has computed. So now we can run those lines here. This will also take a moment, but not as long as the envelope. And so from this, we'll get one list of responses, and that this should contain 20 elements, one for each run, because there are 20 runs for each subject. And each array in this in this list should have the shape samples by channels, and channels are 128, and samples varies a bit between the segments. And then once we have that, we have to do one more thing, which is we have to crop the data to the same length, because usually the recordings are a bit longer than the stimulus. So after the speech stops, the recording goes on for a bit, and so we have to go through there and crop them. I've written it here in the way that you check whatever is longer, the stimulus and the response, and then crop the other one to the duration of the shorter one. It's for almost all subjects, the response is longer than the stimulus. There's like one subject in the data set where the response is like a few samples shorter, so it seems like that recording was stopped prematurely or something like that, which is why I included that statement. And then we normalize the data by z-scoring, which means we subtract the mean and divide by the standard deviation. And that means that then if we would plot the data, the y-axis would tell us for a particular data point how many standard deviations away from the mean that is. And this is just a way to, like a standard way for linear modeling. And it's especially important if you have multiple features, that are on a different scale, because you want to be able to compare the weights across features. But this won't be possible if they have like a widely different scale, because if you have like one feature with much bigger absolute values, the model will assign smaller weights, because uh, because you have basically have to multiply the feature by the weight to get the effect on the response. And if you have like absolutely large features, you have to like make the weight smaller to account for that. And so to be basically have like a fair comparison of all features, you z-score them. Here in this case, it probably wouldn't matter because we're just using a single feature, but just for sakes of like keeping the standard workflow, I included the z-scoring here. So it seems the EG pre-processing has run, so we can do the z-scoring. Okay, so now to the top.
We have a question.
Yeah.
So is this a good time for a question? Camila?
Hi. So with respect to the stimulus and the trials, is this like one single track of all the speech someone heard, or is it cut into different trials, and how long the trials are? And...
Yeah, so it is into, I mean, they're called runs here, right? If you look at the list, it's like the files are called run 1, run 2, run 3. And I didn't record this data, but as far as I know, one run is like one continuous presentation of data. So you hear one run, the speech for one run, then there's a short break. I think people answered some comprehension questions, and then the next one is played. And so I think they're roughly like 2 minutes each. They vary a bit, but yeah, they're like organized in trials. Does it answer your question?
Yes, thanks.
All right. Okay, so now to the time lags. So when we model the relationship between stimulus and response, we have to account for the fact that the response is not instantaneous, meaning it takes, if you hear the stimulus, it takes a certain time until the stuff reaches the cortex and we see something. And to account for this, we compute the TRF computes regression across multiple time lags, and we put the time lag. We do this with a single matrix by sort of stacking time lagged copies of the stimulus. So here I'm using this from mtrf.matrices. I'm using this function lag matrix to generate this time lag matrix X. This is something. So the next few steps is stuff you're usually not doing when you do an actual TRF analysis, but I want to kind of walk you through the sort of nuts and bolts of the method, so you have like a solid understanding of what's happening under the hood. So if we execute this, we'll get a plot here where in the top plot you're seeing the stimulus feature, the envelope, at multiple time lags. Here the blue one at 0, and then the green one is something like 500 milliseconds. And you can see in the beginning there's this, for the yellow or orange and green line, there is this step in the beginning here, and this happens because the time lagged features are padded with zeros, so they're like all of the same length, and they're on the left side, they're padded with zeros. And because we z-scored, the baseline of our envelope is below 0. That is because before z-scoring, the baseline of the envelope is 0, and then if you subtract the mean, it gets shifted a bit. And so if you then pad it with zeros, you're getting this step in the beginning, and this can cause sometimes artifacts at the edges of TRFs. So it's good to know where those come from. And then here we see the neural response in the same time window, and you can see it's pretty noisy, and there's not really any relationship you can just see by eyeballing it. So we have to use statistics to figure out if there's any relationship. now I have a little exercise because I want to keep this interactive and not just talk for 2 hours. And that is so here I plot the first 500 samples of the signal. And what I would like you to do is try to plot the last 500 samples of the time-lagged envelopes. So I'll give you a few moments to figure out how you would do that, and then continue.
My subconscious is so MATLAB trained that I Googled MATLAB index last elements of vector without even noticing.
Okay, I think I don't want to wait too long here because I don't want to just use up all our time for array indexing. And Python has this really cool thing where you can index arrays from the back by putting a minus in front of the index. So I can say here x is the stimulus, the time-lag matrix. So I can say x minus 500 and then just leave the other part here empty, and leaving it empty just says take everything. So this says take everything from 500 samples before the end and then up to the end. And then if we run that, we'll see now here, this is the last samples, and you can see that here now there's no zero padding at this end, but rather the time lag copies are cropped in the end. And this is because there's just we can't quantify the response to the envelope at a later point than the last sample of the unlagged envelope, right? It's just the question, what's the neural response that happens 300 millisecond after this last point of the blue line here, is just something we can't answer because we don't have any brain recording of that time. And so you're just at the other end, it's just cut off. And so this is the time lagged matrix. And I just wanted to take a moment to show you how that looks like. So now, once we have that time lag matrix X, we can compute the ordinary least square regression, which has the closed form solution that's given here, which is the X transposed multiplied by X. And this is called the autocovariance of the stimulus. It kind of tells you how strongly the stimulus is correlated with itself. And then this is inverted, shown here by the -1. And this is then multiplied by the covariance of the stimulus and the response. And the reason that we use this inverse autocovariance of the stimulus is because we want to account for the autocovariance. We can't just consider the covariance of the stimulus and the response, because this is affected biased by the fact that the stimulus is correlated with itself. And so the result of that, this matrix W, those are the weights of the TRF. So this is a matrix that's shaped time lags by channels. Or if you have multiple features, it would actually be a 3D matrix that is features by lags by channels. But since we only have a single one, it's just lags by channels. And those are regression coefficients. So they indicate how a unit change in the stimulus feature is expected to affect the neural response over time. And first, let's take a look at the covariance matrices. So here on the left, you see the autocovariance of the stimulus. This first term here is for the bias term in the regression. So the bias is in linear regression. You have like a plus x times b1 plus x times b2 and so on. So the a is called the intercept or bias term, and then the rest is just your time lags. And you can see there's a diagonal where all the values are 1. And this is just because, of course, every time lag is correlated with itself, is identical to itself. And so the correlation is 1. And then the more you go away from that, the lower the covariance value gets. So this looks kind of like a ridge. If you would look at it from the top and imagine it's like the peaks along the diagonal and then it falls off. And the fact that it's not 0 off the diagonal is indicative of the autocorrelation. And then if we look to the right, we see the covariance of stimulus and response. And you can see there's some, and here it's time lags by channels. And you can see there's difference across channels, but it looks very smeared across the time domain. And that is because due to the autocorrelation, we can't infer like any temporal relationship from this, just from this covariance matrix alone. And this is why we're multiplying it with the inverse of that. And so now we have the matrices and we can run the linear regression. And if we run that code, we get something really horrible, like this kind of spiky looking weird thing. And so why does it look like that? The reason we're getting something like this is because all our time lag, our different predictors, which here are like copies of the same signal at different time lags, are very similar. And so that means that there are a lot of possible solutions to the regression problem, meaning that you could there would be a lot of different distributions of weights across the different time lags that would produce almost equally good solutions, right? It probably doesn't matter if you use like one or the other of two neighboring time lags. And so there's a lot of solutions to the regression fit that are almost equally good. And
sorry, Oli.
Yeah.
Can you explain what we're seeing on the y-axis? So the color is, each line is a different lag.
Oh, no, each line is one channel. I'm sorry. Very good question. Yeah, each line is one EEG channel that's being predicted. And the amplitude shows you the weight of the TRF. So that means how strongly the model thinks that a change in the envelope will affect the response at a given channel at a certain point in time. And so because there are like many different solutions that are equally good, the actual like weight is more strongly determined by the noise than by the brain signal, which is why we see like this spiky looking thing. And this is a problem that's known as multicollinearity. And if you want to understand it more deeply, you can read this blog post. This is a very, very cool blog post that illustrates the problem. But so basically there's a large space of possible solutions, and then the TRF just kind of jumps around in that space. But so to solve the problem of collinearity, we can use regularization, which will here use ridge regularization, which is sort of the standard way of regularizing the TRF. And this simply adds a constant to the autocovariance, which is so here I have this factor lambda and an identity matrix, and that is being added to the autocovariance before it's being inverted. And this has the regularization has the effect that it penalizes large model weights. And so And so this this will like reduce the spikiness, because it will disencourage the TRF to take on those extreme spikes. And so here I use a regularization value of like 10 to the power of 6. And now it looks very different. Now we're getting like much smoother curves. It still doesn't look very informative, but that is also because right here we're only using a I think up here you can see it. I'm only using here one element of the stimulus list, so I'm only using a single trial of stimulus and response. So it's not surprising that we get like a noisy response. So now you can go ahead and try out a few different regularization values, and then just observe how the shape of the functions changes as a function of regularization. I'll give you a moment for that. Yeah.
Earlier in your talk, you said that with higher lambdas, you would get better solutions. So why wouldn't we always use like a high lambda?
Well, it depends. You're not always getting better solutions. There is I mean, depending on your data and how much data you have and how noisy it is, there might be a different optimal value of lambda. So that's a very good question. I would withhold the answer for a bit, because a later section is going to address the question of how to choose the optimal lambda. For now, we can just like play around a bit with it and see how the function changes. And for example, I can set it to something much lower, and then when I set it to something low, it again looks more like the original unregularized TRF. And then when I this was the value I had from the start, and then when I set it to something much higher, like 6 orders of magnitude higher, you can see it's not really changing very much. So it seems like at some point, it's the regularization doesn't affect the shape of the TRF too strongly. The only thing that changes, if you look here, is the scale of the weights. So here you can see it right here. Now we're at a really low scale, meaning the absolute values of the TRF are very small, versus when I had this value here, we get like higher absolute values. So this is so with regularization, we're able to like overcome this problem of multicollinearity. And so this was just supposed to be like a little overview of the steps that happen under the hood when you call the MTRF train function. But so now that we took a look at that, we can just start using actually the MTRF pi functions and make our lives a little easier. So instead of doing all that stuff that I did above, we can just create an instance of the TRF class, and just I call it forward TRF here. And then we can just use the train function to train it on our stimulus and response. Here now we are using the whole data, not just a single trial. So we'll get out a nicer TRF, and then we can plot the result. And we see now we can get a like somewhat nicer looking TRF. And again, here each line is one channel, and you can interpret the amplitude here as the expected change in the neural signal following a unit change in the predictor. So if for example, if we pick out that channel over here, we're expecting based on this model that if the envelope changes from 0 to 1, 200 milliseconds later, we're expecting to see a change of 0.2 at this channel. Although that is not entirely true. That would be true if it's unregularized, but as I shown you before, right? If I increase the regularization, the scale changes. So I can't. Once I start introducing regularization, I can't interpret the amplitude of the weights in absolute terms. So I can't. So that actually my interpretation now is not that I just gave you is not exactly valid, because by regularizing we change the scale. However, I can still relatively compare the weights of the channels. So what you can say is if the envelope changes from 0 to 1, we're expecting a change in the neural signal after 200 milliseconds, and that we're expecting that this change is like roughly twice as big at that channel than it is at that channel, and we're expecting that it is positive at this channel, and we're expecting that it is negative at this channel. So you can compare the magnitude of weights across across different channels and also across different TRFs if they use the same value for regularization.
Two questions. One, the training function uses the whole data set or leaves some of the data out? And the second one is the T-min, the starting time. Should it be like negative so that you can see that there are no effects prior to stimulation?
Yeah, that's actually that's actually what I have here in this exercise. So yeah, feel free to play around a bit with the T-min and the T-max, and then change it a bit, and then maybe think about what would be the appropriate time window for this model. Sorry. I need to get some water. Yeah, sorry, I'm still recovering from COVID, so there will probably be more coughing in this talk.
Feel free to, if you need like a 5-minute break, let us know when there's a good spot for everyone to take a bathroom break or have a 5-minute break.
Oh no, it's okay.
Okay.
So if we increase the T-min, T-max time window, we can see that there seems to be stuff happening before 0, although that is very likely an artifact due to this time step that I showed you before in the time lag matrix. So although maybe there is also some consistent tracking, that would be so. If I would be interested in that, I would. We could also try to do the TRF without zero padding, which means that instead of doing this appending these zeros here, we will just cut off the stimulus at that point. And this way we can avoid this this step function there. And let's see if we run it like that. Huh. Why did that not work? mm. That's weird. Okay, that should have worked, but I'm not entirely sure. I don't want to get into live debugging now. And then if we can also extend it more to the right, the time interval. And we can see that towards the end, if you go like to 0.8 or even like 1 second, make like a really long time interval from -1 to 1 second, we can see that, yeah, here probably after like 400 milliseconds or whatever, there's not too much change going on. So probably like 0 to, or we can also probably something like 0 to 0.4 or whatever would be a good choice here. Any questions so far? You guys want to do a little break, or should we just power through the 2 hours?
1 for break, 0 for no break on the chat. No break is winning.
All right, but maybe then we should still take a very quick break to account for the people who would like a little break. And
sure.
I think I wouldn't mind either. So maybe
yeah.
let's just take like the 4 minutes up to
the
2
hour
and then meet back.
Sounds good.
Great. All right, and let's go on. I'll just skim over this section quickly because I want to continue with the rest of the stuff. So this is just to show you that there's the TRF class has a method to export the data to the M&E evoked class. And so this way, if you have M&E installed, you can do that, and then you can access all the nice stuff for visualizations and so on that M&E offers. For example, what I'm doing here, I'm using this to M&E evoked class, which gives you out a list, and each element of the list is a evoked response, like an ERP, and you're getting one for each feature in the TRF. Here in this case, we only have a single feature, so we're only getting one, but it's still a list. So it's still a list with one element. So we have to take the 0 element of the list to get the actual evoked response. And then we can use this method plot joint here to get like a fancy plot that shows the waveform and the scalp topographies at different points. Okay, so now to this question of how to evaluate and optimize TRFs. And so we already played around a bit with the lambda parameter, but here we're gonna look at how to optimize it, how to choose the optimal lambda. But first, let me show you something else. Here I use the TRF.predict method with the TRF we just trained to get a prediction of the stimulus and an estimate of the correlation between the prediction and the response. So this is also if you call the TRF.predict method and you give it the stimulus and the response, you'll get a prediction and the correlation of the prediction or with the actual response. You can also call predict using just the stimulus, but then you'll only get a prediction and no estimate of the accuracy. And then we can compare it to the actual stimulus. We can compare the response to the actual response and see that it has a correlation of 0.034. And this is the average correlation across channels. So this is the observed EEG in blue and then the predicted EEG in orange. And you can see it looks very different, but apparently it has some correlation. But that is this correlation is actually too high. So this is subject to overfitting, which is because we're training and testing on the same data. So we trained on all of the stimulus and we evaluated on all of the stimulus and response. And so because the fitting the TRF finds the optimal mapping between the stimulus and the response, it will correspond to some of the brain processes that are going on, but it will also just fit some of the noise that's in the data. And so probably we're expecting that if we apply that model to some new unseen data, we probably get a lower accuracy than we would get for the training data. And to overcome this problem of overfitting, we can a cross validation. So this is a function in this stats module called cross-val. And what cross validation does is it splits the data into test and training data, trains on the training data, tests the model on the testing data, and then rotate this around so that each segment is used for testing once. And then we get the average correlation across all these segments. And what we're seeing here, it's like 0.032, which is a bit lower than 0.034. So what I would like to discuss now is, why do you think we see like a rather small change? Could you think of other circumstances where the difference between this cross-validated correlation and the raw correlation up here might be bigger? Like, are there any rules to this? What factors affect the amount of overfitting in a particular model? Anyone has an idea? Like, if we say we would like to increase the amount of overfitting. So there are two main factors that go into this. One is the amount of data that we have, and the other is the amount of parameters in our model. So the more free parameters in our model, the better it is able to just fit random noise in the data. And on the other way around, the less data we have, the more parameters per data point you have for fitting the data. So the amount of overfitting is kind of this function that depends on the relationship between the amount of parameters and the amount of data. Here we only have a single feature, just an envelope, and we have loads of data. Like, I think like it's like almost 40 minutes or something of data. So there's really not a lot of overfitting that can be done by the envelope here, just because there is only a single feature and there's a lot of data. One thing which we could try out is if we increase the number of time lags. And actually I haven't tried this. So we use like 0 to 1 second. We're getting a higher correlation, 0.039. And when we do that on Crosswell, we get 0.037. Okay, so the number of time lags doesn't really seem to affect the amount of overfitting very much. So let's just get back to our value of 0.4. Okay, so this is why one has to use a cross-validation if you want to get a correct estimate of your accuracy. And this can be much more severe in bigger models. Okay, and so now to the question of optimizing lambda. So before I talked about this problem of multicollinearity and how regularization deals with that, but regularization also has another important purpose, and namely it prevents you from overfitting to your training data, and this allows you then to make better predictions about new unseen data. So so this means by regularizing, you're like purposefully underfitting the training data with the aim of making better predictions of new data. So here I've simulated some data, just a little 2D example. So those are just two normally distributed variables that are correlated. So I have the variable x. This is just 100 samples from the random normal distribution. And then y is also just 100 samples from the normal distribution, plus 0.6 times x. So the correlation of those variables should be 0.6. And then we can sample random points from those data, which I do here with this random.choice function. So we pick 5 samples here, and then we fit a linear regression using the same methods that I just described in the beginning. We use the linear regression to fit those sample points, and then see which regularization value actually gives us the closest approximation to the population trend. Here, I mean, in reality, usually we don't know we don't know the whole population, right? We just know the data from our like few participants that we sample from the population. But here in this simulation, we know the actual population trend. So this is kind of the ground truth, is this black dashed line, and then we can see which regularized regression comes closest to that. And here you can see that with like lambda equal 1, maybe lambda equal 2 or something would be even better, but this is the one that gives you the best approximation to the trend based on your training data. So now what I would like to do, since all of this is random, we'll get a different result every time we rerun this cell. So I would like you to rerun it a few times in your own notebooks, and then look at under what circumstances you get a low or high value of lambda will give you the best approximation of the population trend. And then you can also vary the number of samples you draw from the population, so this parameter here, and then see how that affects the optimal value of lambda. So I'll leave a quick break to give you some time to try that out. So do you think there is any rule that you can make out whether, like, in what kind of scenarios a low value of lambda gives you the best approximation of this black dashed line, and in which scenarios a high value works better?
My simulations seem to vary widely from random sampling to random sampling.
Yeah.
So it doesn't seem like I can learn anything, a generalized rule.
So if we try it out, for example, here in this case, we're seeing that the lambda equals 0 regression, so the unregularized regression seems to, or maybe like a very slight amount of regularization, gives you the best fit. And this is because here, those 5 samples we picked, they give they're a very good representation of the overall population trend. And so if that's the case, then of course you don't need any regularization, because your sample is very representative of all the other data. So based on this sample, I will be able to get a pretty good prediction of all of the other data. Well, if I rerun it, for example, like this, now the trend, the raw trend in my sample is much steeper than in the actual population. And so in this case, I can regularize, because what regularization does is it penalizes the weights, so it kind of flattens out the line. Like if you use infinitely large regularization, you'll just get a horizontal line. And so I can flatten it out to get it closer to this population trend. And then of course, it's in some cases, those actually all work pretty well. Damn, I can't get like a really bad one. Sometimes, of course, you're, yeah, maybe like this. Yeah, like sometimes you're, of course, doomed, right? Because if you pick like samples that are completely unrepresentative of the general population, then there is like no amount of statistics that can save you. And this is why it's important to have sufficient sample size, both like on a subject level and population level. So on a subject level, meaning recording sufficiently long EEG from each subject, and then of course recording sufficient number of subjects. So if we can check that effect by changing the number of samples here, maybe from 5 to 10, so double the number of samples. And here generally what we'll see, or maybe let's do it even higher, let's get it up to 20. So now it should be that most of the time, because I'm sampling a large number, most of the time my samples should be representative of the whole population. And so most of the time I should get the best estimate of the population trend with a small or regularization value or no regularization at all. And this is something we're also seeing in TRFs, right? That is that the more data you have, the less regularization you need. Just because you're with more data, you're gonna be able to approximate the general population trend better. Does that make sense? I'm assuming yes. But also what we're seeing here, right, is that you can't really a priori decide what value of lambda works best. Rather, you kind of have to try it out. And this is exactly what we're also doing with TRFs. I mean, in TRFs, we don't, I mean, in actual like data analyses, we don't know our like ground truth population. But what we can do is we can kind of simulate that by just subsampling from our sample, and then fitting on one, and then fitting the model on one part and testing it on the other, and doing that with multiple regularization values, and see under which regularization we're gonna get the best out of sample prediction. And this is what's done if you pass multiple regularization values to TRF.train, which is what I'm doing here. So if we run that, TRF.train will run the cross validation for each value in this array, and then pick the best one to train the final model. And then we can plot the result, and we can see here in this case the value of 26.83 was optimal. The values are spaced a bit weirdly because they're taking along a log spaced axis, but this is generally a good strategy for spacing the values, because the higher the regularization is, the bigger the change in regularization has to be in order to have like the same effect. So increasing from 100 for lambda from 1 to 10 has a bigger effect than increasing it from 10 to 20, and so on. And so now here, this means we're getting the best prediction with lambda equals 26.83. Again, the changes are pretty small. It's like it only goes up from 0.032 to 0.037. That is again because the envelope model is a very simple model. It has few parameters, meaning it is very robust, and we have a lot of data. So our results should not depend that strongly on regularization. But still, there is definitely an effect. And someone asked earlier, like, if like larger regularization is always better, and that is not the case. So here, in this example, and also here in this result, you can clearly see there is like a certain optimum. Like in the beginning, it gets better, and then it gets worse. What you'll see most of the times is that the not here, because again the envelope is such a simple model, but often you'll see like a steeper. I mean, if we would extend the axis, probably you would see it. Maybe if we do it like that. You're usually seeing a more of a change towards the left side. So like in the sense that under regularizing the model has a stronger effect than over regularizing it. And I'm hoping that I can show that to you here. No, apparently this model really doesn't need much regularization. Like often at some point, if you go lower and lower, at some point you'll just see a complete drop off, because you'll end up with essentially this kind of model here, right? But I want to continue now. Yeah.
I have a question regarding the different subjects. So you would try to optimize a lambda for each subject, or would you try to optimize overall subjects, and how do you decide that?
So that depends on what you want to do. So usually you would, if you're like interested in the question of how strongly is that feature reflected in the brain response, so you're interested in that accuracy estimate, that R value, you would definitely do it on a subject by subject basis, because the data of some subjects would be noisier than those of others, and then the noisier data might need more regularization. But there is a problem that if you're interested in directly looking at the weights, for example, I recently like worked on a project where I was comparing the weights of the TRF across different features and across subjects. And if you do that, it's problematic to use different regularization values, because as I showed you here, if you increase the regularization, you're drastically changing the scale of the y-axis, and so it's hard to compare the TRF weights of two different subjects if you use very different regularization. So in this case, I would actually try to find one value that works okay across all subjects, and keep it the same. So it kind of depends on what you want to do in your analysis.
Sorry to follow up. How would you go about doing that manually? You would just pick a few and run it over the different subjects and see what works best for all of them.
So what I did in my project is I just did I optimized lambda for each subject, and then just took the mean of the best lambda, and then took that value for every subject. So for here we would get like 100 now, because I've like changed the spacing a bit, so apparently it arrives at a different value. And then for another subject you get 1,000, and then for another you get 10, and so on, and you just average them, and then you have like the average best lambda, and you using that for every subject. And then it's probably not gonna be perfect, but it's gonna be okay for most subjects at least. Does it answer your question?
Yes, thanks.
And but this optimizing lambda introduces another source of overfitting, because now if we use that R value we're getting out here, if we use that as our final estimate of the model's accuracy, that will be inflated again, because now while we use cross validation, meaning that the TRF weights are only fitted on the train data, the lambda optimization uses both the train and the test data. So we have another free parameter that we're fitting on the on the whole data set. So if we want to overcome that, we have to introduce a second level of cross validation. Which, oh wait, I'm gonna like reset my range of regularization values. And this can be done with this function called nested cross-val here, which is also in the stats module. And what this will do, it will do two nested cross-validation loops. So in the outer loop, you split the data into a train and test set. Then the train set goes into another cross-validation loop where you fit a model using each lambda and then select the best lambda, and then that final model you'll test on the test set that you've split out in the beginning. And then you're gonna rotate through both loops, meaning for each rotation of the outer cross validation loop, you're gonna do like a complete rotation of the inner cross validation loop. And then you're gonna get one R value for each segment in the outer loop, and also one value for the best lambda. And then the mean of this list is an unbiased estimate of the model's accuracy, because the test data was never actually seen by the model fitting procedure. And is the idea of that clear? Why that would be something you would want to do? Okay, but also here again in this case we see that it doesn't make a big difference, again because I guess here we're getting like something like 0.37 as the best result, and this is also what this function spits out. So, and this is again because we can see here the regularization doesn't have a really big effect, right? Most values we're testing are like equally good almost, so it doesn't seem like we're doing a lot of overfitting here. So, and then to finish that section, I want to introduce the topic of statistical inference. Maybe everyone start running this section because it takes a bit. Now we estimated this. Okay, here I had 0.33. I think I used slightly different parameters. Here it's like 0.037, but it doesn't really matter. So let's say we've observed this average correlation between the envelope and the response is 0.033. And now we wonder whether this is a high or a low value. And so we want to do some statistical tests to assess whether this is significant, this finding. And to do this, I already mentioned this in the talk, we can use this function called permutation distribution to randomly permute the stimulus and the response and get the correlation that happens just due to chance when there is no actual relationship between stimulus and response. I think this will run something like 3 minutes or whatever. So from this code, we're getting a histogram, or we're getting values that we can plot as a histogram, where each value represents one random correlation, and then we can compare our actually observed value, which is here the black dashed line, to this distribution to assess the significance of our finding. So maybe could you take a moment and think about how you would now, from this distribution, so here we have 100 values for correlations, how can we calculate the p-value of our actual correlation from this list? Anyone has an idea? I mean, it doesn't need to be the full implementation. It can also just be like conceptually.
Camilla has a suggestion on the chat.
Yeah, this is exactly right. So the so since the p-value is just the probability of the to observe the data under the null hypothesis, we can just count how often that value appears. And in Python, we can just use logical indexing. So we can use R, or a logical operation, R permuted, and we can say where it is greater or equal to, wait, how did I think we said 0.033 or something like that. And what that will give me is just a list of Boolean values, so either true or false for every element. And then I can sum that and divide that by my number of permutations. And this is the p-value, although there's a problem because there's not a single observation. It's 0, but of course the p-value can't be 0. So in this case, so we have to check that. So if it's greater than 0, meaning we have some values, or maybe we'll just call this n. And if n is greater 0, p is n divided by the number of permutations. And if we don't, we can make it a string. And else we can say that p is smaller than 1 divided by n permute. Why did this not work? Oh, I forgot an equal sign here. And now we can print out p. So this means the number of permutations you're using determines how specific your test can be. So if I used 1,000 permutations, I would be able to say p smaller 0.001. But yeah, I guess from this plot, it's pretty obvious that the correlation is not random. So now, okay, we still have about 25 minutes. Now I want to start to look into multivariate models. So far we've only used the envelope. Now I want to introduce another feature, and to just like keep it simple, I'm computing the acoustic envelope, the acoustic onsets, which we get by computing the first derivative of the envelope and then half-wave rectifying that, meaning we're setting everything that is below 0 to 0. So we're only keeping the positive parts. And if we plot that, we can see this is the onsets, the orange line, and so we get positive values wherever there's a rising edge in the envelope. And this is known to be a good predictor of auditory brain activity. Are these like sharp increases in the acoustic input's power? And then we can create a multivariate stimulus matrix by just concatenating our existing stimuli. And the rest works exactly the same. So you do trf.train with your list of regularization values. And then plot the result. I've had an exercise here, but I think for time's sake, we're going to skip that, if that's okay for you. And maybe you can do it later if you want. And that would be to estimate the prediction accuracy using cross-validation of this multivariate model, and then compare that to the univariate envelope TRF. So potentially this one should give us a better prediction because it's able to capture some activity that the envelope model didn't. And what you can see already is that the envelope weights are getting smaller because some of the variance that was formerly explained by the envelope is now captured by this onset feature. One thing that has to be considered with multivariate models is that you're only fitting one value of lambda for both features, and so it might be that you end up with some suboptimal solution because the value for lambda that is optimal for one feature might not be optimal for another. One way to get around that is to use what we call banded ridge regression. And in banded ridge regression, you split your features into different bands and then fit lambda separately for each band. For example, if you had a 16-band spectrogram and 39 phonemes, you could say the 16-band spectrogram is one band and the 39 phonemes are another band, and you're going to optimize lambda separately for them. Here in our case, both bands are just of size 1. The envelope and the onsets we consider a different band. And then we can create our TRF instance and say method equals banded, and this tells the TRF that you want to use banded ridge regression. And the rest is exactly the same, with the addition that you have to provide an argument called bands, and this is the size of the feature bands in your stimulus that you want to fit separate lambdas to. So here in this case, we're going to say 1, 1, because both of our feature bands have size 1, the envelope and the onsets. If, for example, again, my example from just now, if you had a 16-band spectrogram and a 39 and 39 phonemes, you would say 1639 here. Also, you'll get an error message if the sum of those is not equal to the size of your stimulus. And if you run that, this will actually run longer because we're doing an exhaustive search of all possible combination of lambda values, meaning that the more different bands you're introducing, the more different lambdas you're fitting, it increases the computation time exponentially because you're testing all possible combinations of lambdas. So this should be used with some caution. Also, again, now you're introducing more free parameters, so also potentially more overfitting. And then if that has finished running, we can print out the result, and you'll see that there's actually a bit of a difference in the optimal lambda value estimated for both features. Okay, I think I don't want to necessarily wait on the result of this. Are there any questions regarding banded ridge regression? All right, then I want to come to this to topic number 5, which is decoding. So so far we've predicted EEG responses from the envelope, but we can also turn this around and reconstruct the envelope from the brain responses, and that's what's called a backward model or a decoder. And this is simply done by setting the direction parameter of the TRF to -1, and then just run the same model again with stimulus and response. And the TRF will then just figure out that to use them the other way around, the stimulus and the response. This runs a lot longer because the model is a lot bigger. Now our model has 128 features because we have 128 channels. And if you go back all the way to the beginning and look at the autocovariance matrix, so what you see is the autocovariance matrix has the size is square, and the size is the number of time lags times the number of features. Because here's only a single feature, it's only the number of time lags. But now we have 128 features, so we actually have 128 times 35 lags. Oh, it's not running because because it's still stuck on the other computation. But so this autocovariance matrix becomes a lot bigger. So our matrix computations are more demanding now, and this is why the backward model takes a lot longer. Also, the free Google Colab doesn't give us the best CPU. So this takes for a bit, but then once it's finished, we can also plot the result, and we can see now there is actually a much higher correlation, 0.1. Before it was like 0.03 something. You can still, if you plot the reconstructed envelope versus the actual one, there's still not too much you can see just by eye, but the correlation value is a lot higher. However, okay, now it's finished. However, this is also more subject to overfitting because of the higher number of parameters. So if you're gonna, which I'm not doing that here because it would take a lot of time. If we're gonna do the cross validation on the backward model, also I didn't optimize lambda now. But if we did the cross validation here, we would see a much bigger difference between the raw and the cross validated R than we saw with the envelope model. And that is because with all of those parameters, the model can just do a much better job of overfitting the noise in the training data. Another important thing to consider when working with backward models is how to interpret the TRF weights. So that is because the backward TRF does not only amplify activity that is of interest, but also suppresses activity that is not of interest. It kind of acts like a filter to extract this latent stimulus factor from the data. And this means that you can't, so in the forward model, there is like a physiological interpretation. If we go back to maybe the topo plots, there is like a physiological interpretation of the weights, meaning that you can take this distribution of weights to say, well, if there's an increase in the envelope, you're expecting at 130 milliseconds, you're expecting positive deflections in the front of the scalp and negative deflections in the back of the scalp. So you can directly conclude from the model weight to like the physiological activity. Basically, this is kind of like a approximation of an evoked response potential. But this interpretation is not valid when using a decoder, because the decoder does not have the aim of predicting brain responses, but rather extracting the stimulus feature from the brain response. There's a really good paper I linked here that is specifically about this issue on how to interpret, or why you can't interpret decoder weights just in the raw. But one thing you can do, which is a method introduced in that paper, is you can transform those backward weights to a forward model. So there's a method that's called 2-forward, and that will transform the backward model's weights in a way that you can interpret them in the same way you would interpret a forward model. And when we compare that, we see that this like transformed envelope backward TRF is sort of a smoothed out version of the envelope TRF, and this is due to these filtering properties of the backward TRF. So this is what if you want to interpret topography plots of a backward TRF, this is usually what you should do, is this transformation to a forward model. And in this paper, you can read some more detailed reasoning on why this is the case. Any questions on that?
Has that 2-forward functionality always been in the MATLAB implementation of the MTRF toolbox, or is
there
that
is
new?
a similar function there? I think there's something like MTRF transform or whatever. So they definitely also implement that method from this paper. I'm not 100% sure how the function is called. Also, it might be that it was added in the second version of the MTRF toolbox.
Thanks.
Okay, cool. So I think now I can actually, this is the last section, and we have like maybe 10 minutes left. So that works perfectly. Before I start talking, I'll run this code because it also takes a while. So for the last section, I want to address the question of how much data you need. So before, we already saw that the amount of data determines how good your correlations are, how much regularization you need, and so on. But usually when you design an experiment, you have to in the beginning decide on like how much data you're going to acquire from each subject. So let's for now act as if we're in the process of piloting a new experiment, and we want to find out how much data should we collect. And so one way to answer this or address this question would be, you just record a few subjects, like maybe 2 or 3 people, record a lot of data from them, and then like simulate sort of new experiments from this data by splitting this data into short segments and then rerunning your analysis with increasing amounts of data. And then we can see how the prediction accuracy is changing as a function of the amount of data we're putting in. And this is what I'm doing here. So I decided on a segment duration of 5 seconds here. You could also choose something else. And then I'm taking each trial and I'm splitting it into 5-second long bits, so that in the end I'll end up with something like 700 something 5-second trials. And then I can select varying amounts of training data. Here I go up, this is in seconds, so I go up from 30 seconds of data up to the whole data set. And then for each duration, we'll do the whole thing, like fit a model and get a prediction accuracy. One thing is, right at the very beginning, I exclude the last 2 trials for testing. That is because we just need a certain amount of data to get a good estimate of our accuracy. Like if I only have 30 seconds of data and I just tested with cross validation, in the end I'm evaluating on only 5 seconds of data, and this is much too little to get a good estimate of the accuracy. And if you only have like very, very short testing data, you're going to get very extreme results. So on average, you'll probably get the right result, but then in many runs you get like a very large negative number, in other runs you get a very large positive number, and you would have to rerun the analysis many times until you converge on the actual mean value. And so this is why I'm excluding in the beginning 2 segments for testing, and then I'm testing all of those models on the same segments. And then what we can see when we plot the results, so here I plot the correlation coefficient for each training duration and the optimal value of lambda, you can see that the correlation is increasing. So here there's this first step was a bit weird, but there is just some chance involved because we're randomly sampling from the data. And so if I would do such an analysis, if I would actually do such an analysis, I would rerun this whole pipeline like multiple times, like maybe 100 times, and then average, and then we probably wouldn't see something like that. But now here you can nicely see that there seems to be like this sort of logistic curve or like exponential curve here that converges at some limit. And here it seems like maybe after 1,000 or 1,500 seconds, you're not, or maybe even after 500 seconds. So if I would fit a curve, it would probably look something like this. So it seems like we're not getting a lot of extra power in our model just from including ever more data beyond this point. So here I might actually conclude that, okay, getting I don't know, 20 minutes of data from each subject is fine, and that should be enough. Of course, this is only a single subject. And then what you can also see is that as the correlation is increasing, the value of lambda is decreasing. And this is what we've seen before, that with more data, you just need less regularization to fit your model. And do you have questions about that? Should I look in the chat? No. Okay. All right. And then if some of you are interested in doing some more work with this data set and exploring it some more, I have some bonus exercise here for you that you can do on your own. And that is one thing, like if you're in a situation where you can't acquire all the data you want, for example, you're recording from children and you can't just record an hour of EEG from children, this can be to some degree overcome by training, by recording a shorter amount of data from many subjects, and then training a TRF for each subject, and then averaging all the models, and then making predictions with these generalized models. And this has been shown in different papers to improve predictions. And so what one could do is download the other subjects. So you would just use DataLad again, and just instead of the number of the subject, put in an asterisk, and this will download all the subjects. And then we can go through all of the subjects in the same. Oh, let's close this. In the same fashion we're doing it here, we can go through all of the subjects, and then for each training duration, train a TRF for the data of each subject, and then average all the TRFs of all other subjects. Meaning, if you want to predict the data of subject 3, you're going to average the TRFs of all subjects but subject 3. And averaging is really simple with mtrfpi, so you can simply just, like you would with arrays, just add the TRFs and divide them. So an average TRF would work like this: TRF 1 plus 2 plus 3, and then divided by 3, and then predict the data of that subject from the average TRF of all other subjects. And then you could compare the curve of accuracy over time between average and specific models. And so what I would generally expect here is that the average model does better with limited amounts of training data. But then if you have a lot of data, probably the subject specific model should outperform the generalized model, because it's just able to capture some subject specific brain activity that is not present in the average. So something for the interested who want to explore this some more. Also, yeah.
Last question, sorry. Is this one of the cases where you would want to find a regularization parameter for all the subjects, because then you're going to average them?
Yeah, exactly. Here, if you would have one subject that has a much higher lambda than the others, then you would basically ignore that model and the average, because the weights would be so low. Yeah, very good point.
Ole, I have a question on that as well. Thanks very much for this, by the way. It's brilliant and great work on developing the toolbox. So it's recommended not to actually average model weights when trying to combine subjects or even trials or whatever it is, and instead to actually sum over the covariance matrices. And I'm wondering, is there a way using M2F-Pi to do that? Could you input data for multiple subjects in order to generate one model?
I think there is not, no. So I mean, you can of course just use the covariance functions from the toolbox, but that is actually a good idea to do that for the future. Do you know a paper where they looked at the difference of averaging covariance matrices versus averaging weights?
Yeah, there's a paper by Gustafsson 2010 that showed that they're numerically different. And there's a kind of a tutorial on I think it's on specifically on auditory attention decoding models that also talks a little bit about this. I can throw a link into the chat if you want.
Yeah, that would be great. I'll have a look at that. Cool. Yeah, that might actually, I have to think a bit about like how one would implement that to use the covariances, because then you would need to pass in all of the data rather than just the fitted TRFs. But I think it's potentially a useful feature.
Yeah, it is. It also it's kind of a nice way of consolidating a lot of data as well, without having the issues of discontinuities, between trials and things like that. It kind of circumvents all of that, which is
and
really.
Then could you also have different regularization parameters per subject, or you're just
well, that's the thing, you wouldn't if you were to do it this way, you would have to use an individual or just a single regularization parameter.
Yeah,
which might be suboptimal in some ways.
Yeah. All right. Did you share the link to the paper?
I'll just put that in now.
And then I think we should head back to the other meeting room.
Yeah. Cool.
Great. Thank you. All right.
Okay. Thank you so much for such a well-formatted tutorial. There was a lot of embedded papers that people are going to find useful and lots of instructions. So I definitely will be going back to this file later on.
Yeah, great. So happy if it helps.
And thanks for powering through COVID for us.
Yeah, I was a bit
Yeah.
disappointed that the second time COVID was almost as bad as the first time. I thought it would be
Oh.
easier.
Huh. Yeah.
Well, it is what it is.
Yeah. Well, thank you for following through, even though you're sick. Okay, so everyone, if you wish, head over to the other Zoom link. I can try to find it really quick and paste it on here.
Oh yeah, that would be good.
For the Q&A. Okay.
All right.
Bye.